So my background, I'm originally from Belgium, and most of my life I lived in Antwerp, so it's in the northern region of Belgium here. So Belgium is, for those who don't know it, located between France, Germany, Netherlands, and the UK, and it's almost 4,000 miles from Norfolk, where we are approximately are right now, most of you, I guess. And yeah, so here is Antwerp, and here you have the border with the Netherlands, and then here is the North Sea. And I just show some pictures to show you what it looks like, the city. Now here you have the cathedral in downtown Antwerp and our beautiful train station. But what I really wanted to show you is this video that I made some weeks ago when I was in Belgium. And this video was shot from an apartment in the southern part of Antwerp. And what you see here is an overview of the city. And so you have the park here, and here is the Museum of Fine Arts, a lot of bars and restaurants. But why I show this video is because at the start of the video, you may notice this river here. And this is the Scheldt estuary. And it flows the source is in France, it flows through Belgium here, and it passes through Antwerp, and then it will flow into the North Sea. And during my PhD in Belgium, I used to study this estuary. It's called the Scheldt estuary. And this using mechanistic and idealized modeling techniques, as mentioned by Eileen. And I focused on sediment and phytoplankton interactions, so really combining biology and physics. And then 2 years ago, I arrived in the US, and I started working on this very gigantic, beautiful system at Chesapeake Bay, but this time using statistical modeling and focusing on forecasting harmful algal blooms. And the two systems are completely different, although they're both estuaries. Here I show some differences. You may notice that on the video that I showed, you could literally see the houses on the other side of the estuary. So the Scheldt estuary, in Antwerp, it's only like 300 meters the width. Whereas in the Chesapeake Bay, of course, it can go up to 30 miles even. So it's 10 times or even more larger, the Chesapeake compared to the Scheldt estuary. So this is the first important difference. Some other differences, you may notice this turbid color of the Scheldt. The Scheldt is very turbid compared to the Chesapeake. It's almost 10 times and even more than 10 times more turbid if you compare the two systems. And the bathymetry, geometry, the Chesapeake Bay is a deep and non-convergent system. It's more or less a rectangle, whereas the Scheldt is funnel shaped, so the width it converges the more you go land inwards. And then also the discharge is 10 times larger, the Chesapeake Bay, compared to the Scheldt estuary. And what's also very interesting, especially if you're interested in tidal effects, the Scheldt estuary, it has a tidal range that is very large compared to the Chesapeake Bay. In Antwerp, for example, the total depth, it's only like 12 meters on average, but the tidal range is 5 meters and even more. Whereas in the Chesapeake Bay, we talk about 10, 20, 30 centimeters. So this is also a very important difference. Okay, so this is just on my background, what I used to study. Now let's go to harmful algal blooms. Just to be sure that everyone knows what I'm talking about, harmful algal blooms are defined as algal blooms that negatively affect other organisms in natural waters by excreting toxins or severely lowering the oxygen concentrations. So the definition is rather broad. It's not just excreting toxins. And as mentioned by Eileen, recently, why are harmful algal blooms, why are forecasting harmful algal blooms important? As mentioned by Eileen, ODU professors, and some of you are here, they received a 3 million NOAA grant to track harmful algal blooms. So this is just the title of an article that was published, I think, on the ODU website. And then another example why harmful algal blooms and studying harmful algal blooms here are important. Here's another title, Virginia waters troubled by multiple harmful algal blooms. So this was published in the Williamsburg Yorktown Daily, which is a local newspaper near Williamsburg and Yorktown. And they talk about this bloom that occurred in especially the York River. And here I have a video that some of my colleagues, Savannah Mapes, she made this beautiful video of the harmful algal blooms in the Chesapeake Bay, in the York River, in September 2022. You can see these complex patterns there. They correspond to the HAP. And Savannah Mapes is a student with Dr. Kimberly Reese, and they show that the This HAP consists of 3 dominant species, which are listed here. But although it's very beautiful to look at these blooms, they of course also have a harmful effect. And here are just some examples of the potential harmful impact of HAPs. First example is fish mortality, because they may excrete toxins or deplete oxygen concentration, and this may result in fish mortality. A second example is beach closures, because it may be dangerous to swim in waters that are contaminated by HAPs. And the third example is the economic impact of these toxins. They may, for example, accumulate in oysters, which may also be dangerous when you want to eat oysters. So it's therefore important to forecast HAPs and get insight into the mechanisms that drive these blooms. And that's why we constructed the following objective. What we want to do is we want to forecast harmful algal blooms and add this forecast to our tool that we have, and I will talk about it later. It's called CBEFs. And we also want to organize a workshop with stakeholders to improve our forecasting tool. So this is really the main objective when I started working on this project. And basically the idea is to use forecasts that we have that provided by CBEFs. I will introduce this tool later. Forecast of temperature, salinity, and pH, so the environmental conditions. And based on forecasts of the environmental conditions in the Chesapeake Bay, we will forecast bloom probability throughout the bay. And in the end, we want to figure, as shown on the right here, we just want to know where. Yes? Did I get a question or?
No, that.
Okay, sorry, I thought I heard something. Oh, all right. So in the end, we want a map as shown on the right, where you have a probability of HAPs throughout the Chesapeake Bay. Let's start talking about the tool that we have. It's called Chesapeake Bay Environmental Forecast System, or CBEFS in short, and it is like a weather forecast. It's based on the CHESROM. It's called CHESROM's ECB. So it's a mechanistic model based on regional ocean modeling system. So it's a 3-dimensional model, and this model will provide us forecast of the environmental conditions in the Chesapeake Bay, and like temperature, salinity, and other variables. It's a 3-dimensional model. It has a 600-meter by 600-meter model grid and 20 vertical levels, and it resolves the hydrodynamics, like the tides, but also the biogeochemistry, like carbon, nutrients, and other nutrients, and so on, and also phytoplankton. It's forced at its boundaries, land inputs, terrestrial inputs from watershed models and USGS data, but also it's also forced using data at the atmospheric boundary, winds, solar radiance, temperature, precipitation. And it's also forced at its coastal inputs, also using data, observed water levels, and other NOAA data. And this model was developed by my colleagues that are shown here. And if you want to know more about this model, because this was a very brief introduction, I invite you to check out the papers by Beaver et al., so Aaron Beaver, shown here, and Saint-Laurent et al., Pierre, shown here. Okay, we have this tool, we have this forecast. What we also do is we publish these forecasts of the environmental conditions in the bay. We have nowcast and 2-day forecasts. And this is a screenshot of the website where we daily publish our forecast. It's fims.edu/cbefs, and you can also just scan the QR code. And when you go there, you can select a variable of interest, and then you can check out spatial maps, also seasonal trends, and also depth profiles. Let's have a look at some examples. So this is an example for the bottom oxygen. And I just extracted this forecast last week. So on the left, it was January 30th. That was a nowcast. And then on the right, it was a 2-day forecast. So February 1st, last week. And then the colors in this case depict the bottom oxygen. Back then, what we saw is that the waters are pretty healthy. We see high oxygen concentrations throughout the Chesapeake Bay. So it's depicted in blue. But that's because the water is pretty cold right now. In summer, we can have a completely different picture. We typically have hypoxic conditions in the northern part of the Chesapeake Bay. And because we are also interested in trends, we also publish trend plots, forecast trends. Here's just an example, again, for the trend of last week, so the 2-day trend, and then you can just see whether we expect a decrease or increase over these 2 days, in this case, of the oxygen concentrations. Here's another example for salinity. So on the left, again, the nowcast of January 30th for salinity this time. What you, of course, have is that the salinities are higher near the Atlantic compared to the upper reaches where you typically have fresh water. And then on the right, I show the 2-day trend of the forecast, and this is already more interesting. What you see is that in some regions we expected an increase of salinity, whereas in other regions we expected a decrease of the salinity, depicted in blue. We also added some biological indicators, and here is an example for Vibrio vulnificus, again, the nowcast for January 30th. And what we see is that we do not expect Vibrio right now or also last week, just because it's too cold. They typically bloom in warmer waters. And then the trend is also shown on the right. So again, if you're interested, I invite you to check out vims.edu/cbebs. Let's go back to our objective. We want to extend this tool with forecast of harmful algal blooms. And like I said, we're going to use the existing model forecast of temperature, salinity, oxygen, and so on. And we will use this forecast as input in a so-called empirical habitat model. And this model, very briefly, I will talk about it later. It's a statistical model. So you will train the model. It's like your brain. What you do is based on historical data, you train the model. So based on historical information, you train the model. You just compare environmental conditions to whether you detected blooms or not. And then after training, you can apply the model using new forecasts of environmental conditions, and then you can get a forecast of bloom occurrence. That's basically the idea. So if you want to apply such model, you need two things. First of all, training information to train your model, and then also you have to choose a specific statistical model type. More about this later in the talk. Okay, and in this talk, I would like to present some of the challenges that we encountered while developing these models. The first question that we had and we looked at was, how does model prediction skill change in time and space depending on model type and complexity? Like I said, one of the things you have to choose is a statistical model type. So we just tried different model types and we looked at model prediction skill. We started with focusing on percent and minimum. Illustration is shown here. And we published these results in Frontiers in Marine Science almost a year ago. And the second question was not on the effect of model complexity or model type, but was more focusing on the effect of training information, so training data, how this affects the construction of your statistical model. The first question that we had was like, should we use in situ observations to train our statistical model, or should we use mechanistic model output provided by CBEFs to train a model? Because in the end, we're going to apply the model using mechanistic model output. Second, another question was, how do correlations in the training information and in the training data affect your statistical model? And in this presentation, I will focus on question 2, and so question 2A and 2B. And the question 2A, the corresponding results were recently submitted to ecological modeling, and the manuscript is currently in revision. Okay, research question 1. Should we use in situ observations to train our statistical model, or should we use a mechanistic model output to train the model? And because, like I said, we apply the statistical forecasting model using mechanistic model output provided by the CBEST forecasts. So should we also train it using the statistical model using a mechanistic model output, or can we just train it using in situ observations? And why is this important? Because traditionally, if you read the literature, typically statistical models are trained using in situ observations. So that's why we asked ourselves this question. Okay, how can we answer this question? What we did is we came up with 3 model experiments. In experiment 1, we trained the model using in situ observations, and we also applied the model using the statistical model using in situ observations. So that's why we call it method OO, method observations. In the second method, we again train the model using in situ observations, but this time we apply the model using mechanistic model output. So we test it using mechanistic model output. And then in the third method, we train and apply the model using a mechanistic model output. And then finally, we compare the corresponding model prediction skill to see whether we can train a model using in situ observations or whether we should train new models using a mechanistic model output. That's basically the idea. Okay, before we look at the results, I will have to show you the methodology. And like I said, we need two main things. First of all, training information, and then secondly, we have to pick a statistical model type. Let's start with the environmental training information, and let's start with the in situ observations. So these data are provided by the Chesapeake Bay program. It's publicly available data, and we use data from 1985 to 2020. And we have more than, if you do that, you get more than 7,000 data points per taxon because we have one observation each month or every 2 weeks. And we use observations coming from 42 stations, covering both the main channel and the tributaries. And the environmental variables that we focus on are shown here. So it's 9 environmental variables to start with. And we chose these variables because we know they affect blooms, HAPs, and also because of data availability. And these environmental variables, they include physical variables like water temperature, salinity, but also more complex biological variables like total organic nitrogen and dissolved inorganic nitrogen, for example. Okay, then the next step, we also need training information coming from a mechanistic model output. And here, of course, we use the CBEFS data. And what we did is for each observation in situ observed data point, we looked for the corresponding mechanistic model output based on date, depth, and station. Did I get a question or was it just someone? Yeah. Okay, we have training information training data to train our model. What we did is we focused on 7 algal taxa. Most of them are harmful and they're listed here. They're all harmful except Heterocapsa. And also here, the data is provided by the Chesapeake Bay program. But to simplify the problem, we transformed cell counts because people take a sample from the Chesapeake Bay and they go to the lab and they literally count cells. But to simplify the problem, we transformed these cell counts to a binary bloom, no bloom data. And we did that using fixed thresholds, cell count thresholds for each taxon. For example, for percent minimum, we used a cell count threshold of 1,000 cells per milliliter. When we have an observation of below 1,000 cells, we say it's no bloom. When it's above 1,000 cells, we say it's a bloom. And this is based on the literature. Another factor that we really wanted is we wanted to get more generic conclusions. And that's why we focus on 7 algal taxa that cover various seasons and regions. And here I show you the temporal distribution of the 7 taxa we focus on. And indeed, what we see, so the temporal distribution of the number of blooms on the y-axis, and on the x-axis, the 12 months, going from January up to December. And what you indeed see is that some species, they typically bloom in the summer months, such as Microcystis, and then other species, they prefer the late spring months, like Prosentum, April, May, and then other species, they typically bloom in the winter months, like Heterocapsa. And also, when we look at the spatial distribution of the number of blooms, also here we see a variety of habitat preferences. For example, Dinophysis acuminata, it typically blooms in the lower part of the bay, whereas Prosentum typically blooms in the upper part of the bay, and the more extreme case, Microcystis, it blooms in the upper reaches of the tributaries in the freshwater regions. And again, this is important because we want to get conclusions that are more generic. Okay, we have training information. What about the model, statistical model type? We tried to keep things simple, so we started with the so-called generalized linear models. So let me briefly introduce this type of modeling. In the first step, we assume that the binary bloom data follows a binomial distribution. And here I show you the formula for the binomial distribution. It depends on the probability of a bloom, P, the total number of observations, N, and then it will give you the number of successes that are expected, y. And here is the corresponding plot. I just got it from Wikipedia. Let's focus on the red plot here. So let's say you have a probability of bloom of 50%, so 0.5, and a total number of observations of 40. What you expect is that you have approximately 20 blooms. Indeed, near 20, you see this peak in the probability distribution. For example, if you have a probability of a bloom of 50% and you have 40 observations, the probability that 40 out of 40 are blooms is very low. And that's indeed what this distribution shows you. Okay, so that's the first important assumption. Then secondly, what we want is we want to link the probability of bloom P to the environmental conditions, because that's what we want to do in the end. We want to, based on forecasts of the environmental conditions, we want to make predictions of bloom occurrence. How can we do that? The first step is kind of like a funny trick that you apply, but why not? So what we can do is we just first apply the log and then apply the exponential function. It's like doing nothing because the exponential function is the inverse of the log. So when you apply this function, so the identity operator to this formula, you can write it as such. And why do we do that? First of all, because we can generalize this idea. We can write it in what we call the exponential form. We can also apply it when we assume other distributions, not just the binomial, but for, let's say, the Poisson distribution. And that's why it's called the generalized linear model. But what's even more important is that when you do that, you get this variable, what we call eta, which is defined here. It's the log of the probability of a bloom P divided by 1 minus the probability of a bloom P. And why is this an interesting variable? Because if you plot the relationship between the probability of bloom P on the y-axis and this value eta, what you see is that you get the so-called logistic function. When eta increases, you increase the probability of a bloom. And in a generalized linear model, what we do is we assume that eta is related to the environmental conditions using a linear relationship. And so eta is a constant beta 0 plus then a linear combination of your environmental variables, xi, like salinity, temperature, and so on. And then the beta values are just the linear coefficients. And why is this interesting? Because the beta, they have a clear interpretation. For example, when the beta value is negative, positive, let's say, it means that this variable is associated with an increase of bloom probability. When then it would look like this. When it is negative, it means that it's associated to a decrease of bloom probability. So in the end, generalized linear models, they assume the following relationship, and what you do is you're going to fit the beta values to your data, your response, what we call response data. We have no bloom depicted, represented by 0, bloom 1. So here is just an illustration of a potential experiment. And then you're just going to fit this sigmoid curve and this generalized linear model to the data. So in the end, you're just going to fit the beta values and they have a clear interpretation. So that's what we do in a generalized linear model. We assume this linear relationship between the response and also the probability of bloom occurrence and then the environmental conditions. Okay, I introduce you to the training data, the model type, generalized linear models. Then another question that we have that we should answer is how to choose the environmental predictors and the environmental variables that we will use in our generalized linear model when we train a generalized linear model. Because we cannot just use all the 9 variables because this would result in an overfitted model. And we show that we should choose up to 5 environmental variables, xi. And what we did is to determine how which variables to include, we just tried all variable combinations, and then we just picked the best model. And when you have 5 variables, when you can select 5 variables out of a set of 9, you can just apply combination theory and the following formula. You have a total of 381 variable combinations. And next, we just trained 381 generalized linear models, and then we selected the optimal models based on the accuracy in forecasting in bloom and no bloom, and also the so-called ICAR information criterion, or AIC. And a brief introduction to the AIC. What this does, it's a measure between the balance of model complexity on the one hand and the likelihood of the function of the model. So the first term, it's a function of number of model parameters. So it's the complexity of the model. So the higher the number of model parameters, the higher the complexity of the model, the larger the AIC. And then the second term, it's linked to the likelihood function. So how well is the model performing? And this is a negative term. So it means that if the model is better, let's say, your AIC will go down. So the lower the AIC, the better. And here is just one example for, I think it's percent minimum. And here on the y-axis, I show the AIC for these 381 model variable combinations. And on the x-axis, it's just the number of variables included. So the first 9 GLM, they only include one environmental variable, salinity, temperature, and so on. And then in the second tries, we just use up to 2 environmental variables, and then we go up to 5. And in the next step, we pick the best performing models, the best 75% best optimal model combinations. And these are represented by the orange color. So we have approximately 30, I think something like 30, what we call good models. So, and then the optimal variable combination, it corresponds to the lowest AIC, just here. And why is this also interesting? Because in the next step, we can quantify the relative importance of an environmental variable when we are interested in bloom occurrence. And this is shown here. This is quantified by the relative frequency. How did we quantify? How did we estimate this variable? What we did is we just, for each environmental variable, temperature, total organic nitrogen, and so on, we just counted how many times this variable was included in these like orange dots below here, below here. And then we divided by the total number of orange dots, like let's say 30. And so you get a value between 0 and 1. 1 means that this variable was always included was always a good predictor for bloom occurrence. And it's not surprising that temperature has a very high relative frequency, so it's a very important predictor, and this is well known that for when you're interested in harmful algal blooms. And then other variables, for example, dissolved inorganic nitrogen, at least for microcystis, it showed that it was not that important. Okay, so we have training information, we have model type, general linear model. We know how to select the optimal predictors to train our model. So now we can go back to our research question, the first research question on should we train a statistical model using in situ observations, or should we train it using mechanistic model output? And first of all, the main difference between the three methods is the difference in situ observations compared to mechanistic model output provided by CBEVs. So let's first have a look at the comparison between mechanistic model output provided by CBEVs and on the x-axis, and on the y-axis, the in situ observations. The dashed line, it shows you the perfect ratio, and so ideally you want the points to be on this dashed line. And what we see is that for the physical variables, what we see is that we're doing a really good job, and we model the physical variables pretty well. Like salinity and temperature, because they're near this dashed perfect line. The biological variables, they're definitely more difficult to model. For example, total organic nitrogen, what we see is that we typically underestimate the total organic nitrogen concentration. This is depicted by the fact that a lot of points are above the dashed line. And I also show you here the Pearson correlation. And indeed, what we see is that we have a high correlation, almost a perfect correlation of 1 when we focus on salinity and temperature, whereas for total organic nitrogen, for example, we have a significantly lower correlation. Okay, finally, let's have a look at the actual results coming from our 3 modeling experiments. So let's start with, so here on the y-axis, I show the accuracy, and then on the x-axis, I show the 7 taxa that we focus on. And let's start with methods 00, so where we train the statistical model using in situ observations, and also applied it using in situ observations. What we see is that we get accuracies that are in the high 70s, 80s, so which is pretty good, especially given the simplicity of our generalized linear model. But what's interesting is that when we apply these models, so these models trained using in situ observations, apply these models using mechanistic model output, for example, provided by CBEFs for this mechanistic model output, what we see that at least for some species, for some taxa, we see this significant drop in model accuracy when we compare black to blue. This is especially true for Heterocapsa and Microcystis. And this drop in accuracy is pretty significant. It's sometimes more than 10%. But what's even more interesting is that when we train new GLMs, so new statistical models using mechanistic model output, and again apply it using mechanistic model output, and this is depicted by the triangles, we see that we get accuracies which are very similar to the accuracies found when we trained the model using in situ observations, but applied it using also applied it using in situ observations. So the big conclusion here is, should we, the question was, should we use in situ observations to train our statistical model? What we found is that statistical models trained using in situ observations are less accurate when applied to model output. And that was the blue curve. But when we train and apply the statistical model using mechanistic model output, we could enhance model prediction skill. So that's the main conclusion. So we would, if you're interested in model prediction skill, we recommend training the model using mechanistic model output instead of in situ observations. Okay, let's go to research question 2. How do correlations in the training information affect the statistical model training? Let me briefly sketch the problem here. So like I said, we need statistical training information to train our statistical model. But the problem is that often you have correlations in your training variables. You have correlations between, for example, temperature, salinity, solar radiance, and so on. And this is a well-known problem in the literature because it may inflate the statistical variability in your statistical model parameters. And if you change, of course, the statistical model parameters, this may affect the accuracy of your forecasting. So that's why we are interested in this problem. Okay, we used a very similar methodology compared to what I showed earlier. So luckily, I don't have to add many slides on the methodology again. But the big difference is that we added additional environmental variables. Before we only focused on 9 variables, but this time we focus on, in the case of the in situ observations, we include 19 environmental variables that are shown here. And then for the model output, we focused on 15 environmental variables compared to the 9 that we focused on before. And here I show you the correlation analysis. So the color depicts the Pearson correlation. When the color is warm, it's a positive correlation. When it's cold, it's a negative correlation. And to answer our research question, what is the effect of correlations in your training information? What we did is we first train the statistical model using all environmental variables, and then in the second experiment, we train the statistical model after we remove specific correlations in the training information. We used a correlation threshold of 0.4. So when the Pearson correlation is above 0.4 or below -0.4, we will remove one of the 2 variables. And when we remove one of the 2 variables, we have to pick one. So we remove the most complex one, which is typically the biogeochemical variable. Okay, and then on the plot, when we added a black circle, it means that this correlation surpassed our threshold, correlation threshold. And also, what I also have to mention is that within the matching environmental variables between in situ observations and model output are within the dashed triangle. Okay, so let's analyze these results first. So the first thing that we notice, of course, is that we do detect strong correlations in our training information, and depicted by the black circles. And then a second important result is that when you compare in situ observations and model output provided by CBEFs, what we see is that the correlations are luckily more or less the same. But we do definitely have some exceptions. For example, total organic nitrogen and salinity, it shows a negative correlation in the in situ observations, whereas we do not detect this negative correlation when we look at the mechanistic model output. Okay, let's have a look at the results to answer our research question and to answer the question, what is the effect of correlations in your training information? And that's shown here, but I need some time to explain these graphs because these graphs contain a lot of information, a lot of results. On the y-axis, we have the 19 environmental variables. This time we focus on statistical model training using in situ observations. And on the x-axis, we have the 7 algal taxa. Then the color depicts what we call the association with bloom probability. When the color is red, it means that the environmental variable is associated with an increase of bloom probability. When it's blue, it means that the environmental variable is associated with a decrease of bloom probability. For example, temperature, we see that for most taxa, we have a negative association between temperature and blooms. Only for microcystis, we see a positive association, and that's just because microcystis, like I said, typically blooms in summer months. So the higher the temperature, the higher the probability. And by the way, the association, what we call association, it's just linked to the sign of the beta value. So we train multiple models and we just checked what is the sign of the beta value. And like I said, when the beta value is positive, it means that the environmental variable is associated with an increase of bloom probability. When it's negative, it's associated with a decrease in bloom probability. We train multiple models and we just check like how many times do we get a positive or negative sign. And so in the extreme case, when it's 1, it's always positive, the corresponding beta value. When it's -1, it's always negative. So that's how we computed the association. Okay, let's go back to the results. And so the left figure shows you the results, so the associations without removing correlations in the training data. So we just used all the environmental variables. Whereas in the second plot here on the right, we removed correlations, and the variables that were removed are indicated by the crossed out rows. So we removed total organic nitrogen, pH, solar radiance, and so on. Okay, when we compare left and right, we see 3 main patterns. First of all, when we do detect a strong association between an environmental variable and bloom occurrence before removing correlations, so when the color is blue or red on the left, we also detect typically the same association on the right. So when it's blue here, it stays blue. When it's red, it stays red. That's the first important result. Then the second important result is that sometimes we do not detect a strong association between an environmental variable and bloom probability before removing the correlations. But after removing the correlations, we do detect a strong association. For example, for salinity and heterocapsa here, we did not detect a strong association, but after removing the correlation, we do detect a strong negative association. Okay, so that's also a very important result. So it means that, like I said in my title, correlation in the environmental training information hinder the detection of key environmental predictors. And then a third important result is that the best, really literally the best environmental predictors that we could use to train our statistical model, it also depends on the training information, on whether we remove correlations. And this is depicted by the triangles. And the triangles, they show you the best choice in environmental predictors when you want to have a high model accuracy. And you indeed see that the optimal predictors, the triangles are different left and right. Okay, so in the previous slide, I showed you the results when we trained the statistical model using in situ observations. And here I show you the results when we use mechanistic model output to train our model. And we see a similar trends as I showed before. But what's important is also that the training using mechanistic model output may result in different key environmental predictors. And this is if you compare the triangles from this plot to the triangles from this plot. But like as before, when the color is blue or red on the left, it stays blue or red on the right typically. And also sometimes we do detect additional strong associations when we remove correlations. For example, here for pseudonychia and salinity, we did not detect a strong association, whereas after removing correlation, we do detect a strong negative association. Okay. And finally, final results. Let's have a look at the corresponding model skill. So what I show here is the accuracy of forecasting a bloom on the left, and then the accuracy of forecasting a no bloom occurrence on the right, for again the 7 algal taxa. And on the top, I show the results when we use in situ observations to train our statistical model, and then at the bottom, I show the results when we use mechanistic model output provided by CBEFs to train our statistical model. And I think the most important result, first I also, what I have to mention is that I show the results when using a correlation threshold of 0.4 as done before, but I also show the results when I use a different correlation threshold of 0.5, 0.6, 0.7, and 0.8. And I think the main result, the main conclusion here is that removing correlations can result in a reduction, a lowering of your model accuracy. For example, for these two taxa, procentaminim heterocapsa, we see that when we implement the low correlation threshold, so when we remove more and more environmental predictors in our training information, this may result in a lowering of your model accuracy, depicted by these blue dots when you compare them to the other dots. But this also clearly depends on the taxon of interest, but it also depends on whether you use in situ versus mechanistic model output to train your model. And indeed, we do not see this drop in accuracy when we train a model using mechanistic model output. So it's pretty complex. And here are the conclusions. How do correlations in the training information affect statistical model? The selection of the essential environmental predictors and their association with species presence not only rely on the intrinsic biological characteristics of the species dynamics, so when we compare the various taxa, we saw different results, but it also depends on the training information, whether we remove correlations and whether we use in situ observations or mechanistic model output to train our statistical models. So it's pretty complex. And why are these results important? Of course, we use these insights to construct our forecast, and this is just one example for percentile minimum. So this were some of the results of the first paper that we published. On the right, you have the 5 environmental predictors for percentile minimum in this case. So it's temperature, solar radiance, total organic nitrogen, salinity, and pH. And these are mechanistic model hindcasts for 2014. And then we use these hindcasts as input in our empirical habitat model to get the forecast as shown on the right. And in this case, we used a so-called generalized additive model. This is a more complex model. It's very similar to generalized linear model, but instead of linear coefficients, the beta values, you allow for non-linear relationships. But what's cool here is that we get these like seasonal trends that comply with the observations. So typically you have a bloom in May. Now it's May in the upper region of the bay, depicted by in red. So and then it typically goes down when you have the summer months. So the probability will go down. You have more blue. Okay, and then also we started publishing our forecast on the website vims.edu/cbefs. Here's just one example of a forecast, a nowcast April 14, 2023, and then the forecast April 16, 2-day forecast. And indeed, what we see is that we get these high probabilities in the upper reaches of the bay, which of course complies with the data. And finally, I would like to acknowledge Suzanne and Karen's work because they do a great job in organizing workshops with stakeholders, and this to improve our tool based on the stakeholders' needs. And I just want to mention one specific example. At some point, I attended a workshop with anglers from Maryland and Virginia, and they really showed interest in our tool because they said it may help them deciding where to go fishing. So it's really economic motivation, but it may also help them in avoiding HAPs because it can be dangerous, of course. One angler, they said that at some point they got a severe infection because of a harmful algal bloom. So that was also pretty interesting to hear. But they may also help us in improving the tool because Suzanne and Karen did a great job in asking many questions on what kind of visualization do you prefer? Do you like the color maps? Do you prefer surface plots and so things like that? What kind of regions or seasons are interesting? So I definitely want to acknowledge their work as well. And then take-home messages. This is my last slide. We looked at type of training information, how this affects the construction of statistical habitat suitability models. And we mainly focused on two aspects. What is the effect of training your statistical model using in situ observations versus mechanistic model output? And what is the effect of correlations in your environmental training information? How does this affect your statistical model?